If I asked you to name the most dangerous animal in the ocean, what would you guess? You might be surprised to hear it isn't a shark. It's a flimsy animal, almost clear, 600 million years old, called the box jelly. They look like plastic bags, but they contain potent poisons that are the most dangerous in the world. I work here in the lush tropics at the University of Hawaii in Oahu. My name is Angel Yanagihara, and I'm a research scientist. For the past 17 years, I've been studying one of the world's most fascinating creatures, the box jellyfish. One species I study is called Alatina. They have an extremely nasty sting that can put people in the hospital. I think of them as night owls of the sea. These silent deep-sea hunters swarm shallow reefs to spawn in the darkest hours of the lunar cycle. I collect them right here, in famous Waikiki Beach, about 9 days after each full moon. We've long thought that this was a phenomenon unique to this species in Hawaii, but maybe not. I was pretty excited then when I got a call from somewhere far away asking if I would come and have a look because they thought they have box jellies. In fact, they think they have the very same species that we have here. My mission is to find out if the species I study in Hawaii also lives on the other side of the planet. To determine identity, I will need to observe their behavior and collect DNA. From Hawaii, I fly to Bonaire. It's in the Caribbean, almost to the coast of Venezuela. On the surface, it's hot and dry, a desolate but enchanting island that seems stuck in time, with many vestiges of colonial 16th century Spain. Underwater, it's spectacular, an equatorial ecosystem with mind-boggling biodiversity. And this is where our expedition takes us, into the tropical waters in search of box jellies. What species are here? How dangerous are they? And how do they reproduce? This dive site is called Oil Slick Leap. Has a nice walk down and a pretty steep drop off. Interestingly, a few years ago, some Dutch researchers here discovered potentially a new species of jellyfish. We're going to come back here after dark and dive and look for samples. The species found by the Dutch may be the same as we have in Hawaii. To find out for sure, we need to collect some and fully document their genetics. With a bit of luck, we'll collect animals attracted to our lights and see if they are coming in to breed like the species in Hawaii. If we can record the actual life cycle, our research will make history. Finally, voila, our first box jellyfish. Then another, and more. We're right along the main road. It's the 9th day after the full moon, and we're finding box jellies coming in along this shore, just as we do in Honolulu. These animals can see where they're going because they have eyes. Like the jellyfish we've seen under the microscope in Hawaii, we can count not just 2 eyes like us, but 24. So their behavior is identical to the box jellies in Hawaii, half a world away. Now we need DNA to see if it's actually the same species, and if so, how did they get here? it's often the case in science that answering one question can create many others. Some children here are interested in the work. Let's see what they wonder about.
How long can a box jellyfish grow?
What do they eat?
How do jellyfish get babies?
To find the answers to these questions and many more, we head to the lab.
Bonaire.
Last night we caught a number of adult animals and we have them in the tank here. We've been watching them through the night, and this morning it's clear that they have developed embryo strands. Those strands within the bell there are fertilized eggs. We took the strands that we saw falling out of the adult animals and we put them under this dissecting microscope. Here we can see that the strands are filled with eggs. And when we go at higher power, we can see that the eggs are actually starting cell division, which indicates that they're embryos, they're fertilized eggs. We put some of those eggs under the high power microscope and have been taking images of the cell division as it occurs at higher magnification as well. We then see a world first. Our fertilized eggs have developed into spinning organisms with red pigmented eye spots. After some 30 hours, they stop swimming, settle into one spot, and begin growing vertically into polyps that eat microscopic plankton. When big enough, they'll start swimming again. One day, they will catch and eat fish, which they can kill with a surprisingly savage attack. So what makes a box jellyfish so dangerous? Two things. First, how it venomates. Under the microscope, we see tiny little dots on a tentacle. These are stinging cells. Only in slow motion can we see what may be the fastest motion in the animal kingdom. Thousands of microscopic tubules shooting out like hypodermic needles. This is a model of a nematocyst, or a stinging cell. And basically, these tiny capsules are microscopic. What happens when the tentacle stings is thousands of these stinging cells explode and come out of the nematocyst like that, these tubules, and this is what penetrates your tissue. That's what goes inside your skin. This leading edge breaks off, called the lancet, and this long hollow tubule is stuck in your arm or wherever you're stung, thousands of them. The capsule also is lost, but the venom inside the capsule is first pushed out into your deep skin. So this kind of a long tubule, in fact it's far longer than this model shows, is covered with tiny spines that act like little fish hooks that keep that tubule stuck in your arm so it doesn't pull out. Under even higher magnification, we can see how each tiny spear has a harpoon-like tip. The second thing that makes the box jelly so dangerous is the nature of the venom itself. Through these spears, it pumps deadly toxin. We take the animals back to the lab and cut the tentacles off. Then we remove the stinging cells from the tentacles using this cold solution approach. And basically we rotate them in at 4 degrees at refrigerator temperature, and the little tiny stinging cells come off intact from the tentacles. We then purify those stinging cells, break them open, and isolate the venom. We look at the activity of the venom through different bioassays. One of these assays involves human blood, and we look to see, is the venom strong enough to cause what's called hemolysis, or rupture of the red blood cells. And what you can see here is a plate, and the red wells symbolize that the red blood cells have been broken. We can measure the amount of hemolysis that occurs in each well over a period of time, and that relative to the concentration of venom gives us what's called a specific activity. So we could determine how strong one venom prep is to another, or venom from, say, a sea anemone versus a box jelly is. And we can also look at ways to slow down the venom effect by adding inhibitors to this experiment. We discovered that the toxinology of the Bonaire species is the same as that of the box jellyfish in Hawaii. They both belong to a family of jellyfish with the deadliest venom on the planet. We've answered our key questions. The Bonaire species is the same as the Honolulu species. Their reproductive behavior and attack methods are identical. But this brings up even more questions. How can they be the same? Identical species on different sides of the planet? How long have they been separated? A decade? A millennium? Maybe a million years? And for certain, once we've answered those questions, there will be more. That is the nature of science. The work we are starting here will eventually be passed on to a whole new generation. What color is that? I think the basic ingredient of a good scientist is a child that never grew up. I can remember, like most kids, being 3, 4, 5 and being consumed with the question of why. So I think most everyone is at their core a scientist. Well, I was stung myself in 1997 when I was swimming offshore at dawn, and a woman warned me not to go swimming because there were jellyfish on the beach, but I thought that would be just kind of silly. It wasn't a big threat. When I was stung, though, it was incredibly painful, and I had a very hard time even making it back to the shore. And when I finally did get to the shore, I collapsed, and the next thing I knew, I came to in an ambulance. So it certainly was a lot more than I had expected. The pain lasted for days, and it was this absolutely horrific burning pain. And after that, then I had another 12 weeks of this raised rash, itchy rash like poison ivy. So after that first 3 days of complete agony, I was curious what's in the venom. So I did a literature research to find out what had been published in this field, and basically very little had been done. So as a biochemist, I thought this would be something interesting to look at. So I wrote a grant proposal to do that, and that's what started my whole 17 years path looking at these animals. Looking at these almost clear blobs, it's hard to imagine that they have much sophisticated anatomy, but they do in fact. These animals are in formaldehyde, so they've turned a little bit white. The live animals that we'll be seeing later, you'll notice, are almost completely clear, but they do have a very sophisticated anatomy. They have eyes, in fact 24 eyes, and 8 of their eyes are as sophisticated as our own, with an iris and a retina. They have a stomach, they have a mouth up here, and they have gastric cirri, the digestive organs, up at the top. They also have these remarkable tentacles. In this case, this animal has lost the tentacles, but they are up to 3 feet long, and they are filled with tiny stinging cells, which contain venom. So the final part of the animal, which is really cool to notice, are the gonads. They have 4 long leaflet gonads on each of the corners of the animal, and we'll see that in more detail later. That's a great question. A lot of people watch the television and get their ideas of how to deal with a jellyfish sting from TV, like the episode of Friends where they showed somebody peeing on the poor person who'd been stung. Now, while it's kind of funny, a lot of people think that perhaps that's a good way to go. There is some reason for this to have some potential effect, and that is basically that the venoms are built to work in cold water in the sea. So putting the site where you've been stung in any kind of hot water environment will help slow the venom, and in some cases irreversibly inactivate the venom. Also, pee, urine, has a low pH, and certain things cannot work at low pHs. So that also helps, but a better substitute for that would be vinegar, a spray bottle of vinegar. But that said, putting a low pH vinegar on there only buys you a little time. It doesn't inactivate the venom permanently, and it doesn't remove it from your skin. So a long soak in hot water at this point is a good idea. My lab work has resulted in the development of a novel treatment, and this has been the subject of multiple patent applications and license. So hopefully this will come to the market soon. I have it formulated here for swimmers, for long distance swimmers, in a lanolin base, so it's very sticky. The type that would be available to the beachgoers wouldn't be this type of sticky formulation, but another formulation. So basically, though, we have been able to find out how to inhibit the action of the venom and to stop that in its tracks. So far superior to just a spray bottle of vinegar or the old school idea of pee. I never imagined all the things that I've done over the last 17 years would result from that original sting. But first of all, I wrote that grant, which was funded to do the biochemistry. But in order to do the biochemistry, I had to go out in the ocean to get enough of the animals. Eventually that led me to getting certified as a scientific diver and advanced scuba training, and that also led me to understanding more about the actual life cycle of the animals. But the ultimate benefit of all my work is determining how to stop the venom. And having developed this treatment, I then was contacted by various folks around the world. One of them, Diana Nyad, a famous swimmer, attempting to swim from Cuba to Florida, and she'd been stopped multiple times in the past because of jellyfish stings. So she invited me to come along with them and actually be on the boat immediately tending her all night long, every night of her swim, when box jellies came up by the thousands. So that was pretty interesting, and the treatment worked wonderfully. The physicians on board were really excited by that and wrote a paper in the Journal of Anesthesiology about this work. This work has also been published in open access journal PLOS. Other cool things that have happened, I was invited to be a part of a couple of different documentaries, Nova, Nat Geo, Discovery Channel, even a French documentary was made and included our work. So it's been a long, interesting path, and all started from being stung myself. So you never know what things that seem to be horrible at the time can lead you to amazing discoveries and a whole another kind of career approach and path. My advice is to stick with it, to do the hard work, to get disciplined, get motivated, just go through the process. Learning the basics of math and chemistry, biochemistry, biology, all these textbooks that you are presented with are basically the keys to the kingdom. It's like getting your driver's license. Once you learn all these basic things, you have what it takes to create new technologies, to get new ideas. In order to get there, though, you do, just like a driver's license, need to practice and learn and study. And each of those steps, while they might seem at the time boring or tedious, they lead you to a place where we don't have enough young people. We need more people in the sciences. We need more people, science, math, engineering, especially, young American kids get wowed into other fields, and I want to stress how exciting basic research can be and how exciting science is. And the way to get there really is this old-fashioned paved road of mastering the fundamentals. So get excited, have a dream, hold on that dream, be perseverant, and then be disciplined. Just keep on marching up that hill. It definitely is worthwhile. I think I define science as the basic mindset of asking why. And I can remember, like most kids, being 3, 4, 5 and being consumed with the question of why. And that basic curiosity, I think, is probably one of my most enjoyable kinds of pastimes, is to really kind of consider what's going on and why. And as a little kid in Alaska, I went along with my dad who was fishing, and I used to watch the starfish along the granite walls of the beach there and wonder why they were the colors they were, why they were where they were, and I drew little pictures of them. I think for most folks that passionate curiosity and love of nature somehow gets lost along the way. I'd like to see more young people keep that alive, follow that up, and really enjoy that concept of following why. And that curiosity is a great path. And I think basically human beings are sort of wired that way and built that way. So I think most everyone is at their core a scientist. Now, how do you get to be a research scientist with a profession in research science? Well, that is a long road, a particular path, but it's not quite different from like learning to drive and other things folks do in life. So, but in order to be able to do research as a career, there needs to be a disciplined, long-term, passionate dedication to following that dream of being able to ask why. But at the end of all that training, I would encourage folks that there's a fantastic playground basically waiting to be explored, and we need more people to help do this important work.